So, hi everyone, my name is Rodrigo Ochoa. I am part of the Biophysics of Tropical Disease Maxwell Tandem Group. And today I'm going to present this poster that is the computational design of peptides bound to the MHC class 2. This work has been developed also together with International School for Advanced Study, CISA at Trieste, Italy. So what I'm going to do is I'm going to present different sections of this poster showing the main results and the methodology that we applied for this work. So basically the main motivation of this work is a design protocol that we have been working for several years. And the idea is to apply it with the MHC class 2, that is a very important protein target related with the immunological response. So in this case, we are starting from a protein peptide template of the MHC class 2 bound to a peptide. And the protocol is a stochastic search that basically we select a residue in the peptide and we perform a single permutation. Then what we do is that we sample the protein peptide system using molecular dynamics with explicit solvent. And then we score the final system by using scoring functions and scoring different frames of the trajectory. With that information, we apply our criterion to accept or reject the mutation. And based on that, we update or not the sequence, and we start again the protocol. So for the scoring strategy, we are using 6 different scoring functions based on a previous benchmark of this particular system. And for the acceptance, we are using a consensus metric where, for example, if 3 or more of the 6 scoring functions are in agreement, the mutation is accepted, otherwise is rejected. And to evaluate the progress, what we do is that we start from an initial structure of the MHC class 2. And then what we do is that we start a number of attempted mutations. So in our case, we attempted a total of 100 mutations. And based on the attempted mutations, we follow up the scoring function and we see if the values start to get minimized after these iterations. So basically, we did this strategy different times, depending on the criterion to select or not a particular residue modification. So I'm going to explain that in the next sections. So initially, before starting the design protocols, what we did is sample the system during 100 nanoseconds. This is just to check convergence of the system, to see if the bound conformation is stable. As you can see here, I am highlighting 4 particular positions on the peptide that are crucial during the binding affinity. And also we found that despite the peptide is linear, the conformation is very stable according to the RMSD, and also hydrogen bonds between the peptide and the protein. So because of that, we use the last frame to start the protocol. And for the protocol, the initial strategy was to perform, for example, random mutations on the peptide sequence, and then follow up the scoring functions as I showed before. So what I'm showing in this plot are basically the scoring functions for during the 100 mutation attempts after doing these random modifications. As you can see, for many of the scoring functions, the values are going down. But in some cases, we can find fluctuations like this one here. So this is important to notice that could happen because of the consensus strategy. So the idea is to explore in an optimal way the sequence space. And in this case, for example, we found that 20 to 30% of the sequences were accepted, so which is good for the acceptance rate of the protocol. However, one of the main issues about this strategy is that some of the peptide sequences tend to be a little bit hydrophobic. So because of that, we decided to use other strategies to select the mutations. So we applied different additional strategies. One of them involves the use of bioinformatics filters to, for example, avoid hydrophobic residues. so very hydrophobic peptide sequences. And the other strategy is because this system has been studied a lot, so we have information of peptide binders and the frequency of amino acids on these particular regions. So we decided to use another strategy, selecting amino acids that has a higher probability to be part of these peptide binders in this particular system. So we found a lower number of mutations accepted. However, the peptides have better properties and are also good candidates for the design of these new modifications and for experimental validations. So basically, after doing the different strategies, we obtain a final list of 105 peptide candidates that you can see plotted here in this figure. Here, what we did is we applied a hard filter to pre-select some of the mutations based on the bioinformatics properties, some empirical rules, and also based on an average ranking using all the scoring functions we applied before. And then what we did is with the 25 peptides that we selected, we run longer ND simulation of 200 nanoseconds. In this case, to do a similar average ranking, but using more trajectory information. And we did a new ranking, and we selected 12 peptides that are currently in experimental validation. So just as a conclusion, basically the protocol aims to predict in a computational efficient manner new peptides bound to a particular target. And basically, in the case of MHC class 2, could provide a complementary approach to other knowledge base and machine learning methodologies to include a little bit a de novo characteristic in the prediction of affinities for peptide bound to the system. So here there are some references. In the poster you can find more information. And if you have any questions about it, just let me know. And thank you very much for your attention.